Diffusion Mobile, quality e-learning experience on the go.
Yo, welcome to yet another biological session. I will be my humble self, your tutor for today. I am Oshutogun Olajide, aka Smoking Brain, and today you and I are going to be looking at diffusion. Now, don't forget, this program is made possible by Fusion Mobile e-learning platform, solution clinic, so to speak. So today we're going to be looking at these outlines. We're going to be looking at what is diffusion, the factors affecting diffusion. We're going to be looking at osmosis, the differences between diffusion and osmosis, D and O, diffusion and osmosis. We're going to be looking at plasmolysis. We're going to be looking at hemolysis. We're going to be looking at the differences between plasmolysis and hemolysis. We're going to be looking at turgidity, and lastly, we would be looking at flaccidity. So the question at hand is this: what is diffusion? What is diffusion? Have you ever sprayed a perfume before in your room? And you would notice that the point of spray is higher in concentration. You will notice that the perfume smells greatly at that particular point where it is being sprayed than the other end of the room. So basically, what is diffusion in that sense? Diffusion is basically the movement of ions. Diffusion is basically the movement of ions or molecules. Diffusion is basically the movement of ions or molecules from a region of higher concentration, to a region of what? Lower concentration. The movement of ions or molecules from a region of higher concentration to a region of lower concentration until when? Until they are evenly distributed. And the word evenly talks about what? Equality. Until they are evenly distributed. And so now can we just try and break down these strange words into bits? I said diffusion is basically the movement. Now movement means our ability to change position, ability to translocate, ability to change a place. And that is movement. Now the movement of what? The movement of ions or molecules. What are ions? Now ions are atoms or groups of atoms that are positively or negatively charged. Again, ions are atoms or group of atoms that are charged. Now they are either positively charged or negatively charged. Now what are molecules? Molecules are atoms or group of atoms, so to speak. So when ions or molecules are moved from a region of higher concentration, a region where they are greatly felt, to a region of lower concentration, a region where they are not felt at a particular time, until they are evenly distributed, we say that is what? Diffusion. Again, diffusion is a movement of ions or molecules from a region of higher concentration to a region of lower concentration until they are evenly what? Distributed. So now let's look at diffusion. Let's look at diffusion as a particular act, as an act, so to speak. Now diffusion takes place in liquid and gases. Diffusion takes place in liquid and gases. Diffusion takes place in liquid and gases. Now, what are the factors that might tend to affect diffusion? Can we just look at it? Factors affecting diffusion. Factors affecting diffusion. Factors affecting diffusion. The following are the factors affecting diffusion. Number one, the nature of matter. The nature of matter. The nature of matter. Number two, concentration. Nature of matter, concentration. Number three, temperature. Now that we've got these three lines, we just talk on the nature of matter. Now matter exists in three states. Matter exists in three states. We have solid, we have liquid, and we have gases. Now when it comes to diffusion, diffusion is greatly felt in gases than in liquid. Why? Because the molecules of gases are not tightly packed. What do I mean by that? gases have free mobile electrons. The electrons are allowed to move about. They are allowed to collide with one another and with the wall of the container. Liquid have electrons that move about, but they are restricted up to a particular point compared to like solid. So diffusion is greatly felt in gases than in liquid, and then compared to solid. Diffusion is great in gases compared to liquid and solid. Diffusion is great in gases compared to liquid and what? Solid. Now when it comes to concentration, concentration simply talks about the volume or the amount of substances. Now the higher the concentration, the greater the rate of diffusion. Again, the higher the concentration, the greater the rate of diffusion. The lower the concentration, the lower the rate of diffusion. Now lastly, temperature. Temperature affects diffusion greatly. As the temperature is increasing, diffusion is what? Increasing. The higher the temperature, the higher the rate of diffusion. The lower the temperature, the lower the rate of what? Diffusion. Are we there? The higher the temperature, the higher the rate of diffusion. The lower the temperature, the lower the rate of diffusion. And what do we have here? The higher the concentration, the higher the rate of diffusion. The lower the concentration, the lower the rate of diffusion. Now diffusion is greatly felt in gases than in liquid, and liquid compared to what? Solid. And lastly, we have what we call nature. Lastly, we have what we call molecular size. That's the fourth one. Molecular size. What we call molecular size. Molecular size. Now it is sufficient to know that the greater the size, the lower the rate of diffusion. Are you with me? The greater the size, the lower the rate of diffusion. The smaller the size, the greater the rate of diffusion. So whenever we are talking about molecular size, we are talking about an inverse relationship. The relationship is not direct. The relationship is not direct. We are talking about an inverse relationship. As the molecular size is increasing, are you with me? The diffusion is decreasing. As molecular size is increasing, diffusion is decreasing. As molecular size is decreasing, diffusion is increasing. Are you with me? So that is what you need to know. Again, what are the factors affecting diffusion? Number one, nature of matter. And number two, we have molecular size. Number three, we have concentration. And number four, what we call temperature. Now, as concentration is increasing, diffusion is increasing, and vice versa. That means the other way around. As temperature is increasing, diffusion is increasing, and vice versa. Diffusion is greatly felt in gases compared to solid and liquid. And lastly, for molecular size, the greater the size of the molecule, the lesser the rate of diffusion. So now, basically, we're looking at the word osmosis. 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 Now, what are osmosis? What are osmosis? What are osmosis? That's what we're going to be looking at. So I need you to concentrate. I need you to pay attention. Now, osmosis is the movement of water molecules specifically. Are you here? Osmosis is the movement of water molecules. Osmosis is the movement of water molecules. Specifically, they deal with water molecules. Whenever you hear the word osmosis, what must come to your mind is water molecules. It's the movement of water molecules from a region of what? Lower concentration. The movement of water molecules from a region of lower concentration to where? To a region of higher concentration. From a region of lower concentration to a region of what? Higher concentration. Now, for osmosis to be carried out, it needs a particular medium. It needs a medium. It needs a medium. Now, are you with me? A medium simply talks about a material, so to speak. Now, it is the movement of water molecules from a region of lower concentration to a region of higher concentration through a semi-permeable membrane. Through a semi-permeable membrane. Are you with me? Osmosis is the movement of water molecules from a region of what? Lower concentration to a region of higher concentration through a selective or a semi-permeable membrane. Now, what are water molecules? You and I know water exists in three states: when water is solid, we call it ice, and when water is liquid, it is also known as your normal water. Now when water is gaseous, we call it steam. So here water molecule, we're talking about water as in liquid, not water we're talking about water here basically in normal water form, not water in gaseous form. So when water is moved from a region of lower concentration to a region of higher concentration through a semi-permeable membrane. Are you with me? We say such movement is what? Osmosis. Now what is a semi-permeable membrane? A semi-permeable membrane is any membrane, any material that selectively allow the passage of materials, that selectively allow the passage of water, that selectively allow. The word selectively talks about the fact that it will allow some substances or material to pass and it will not allow some others to pass. So it is selective. It has fan. Are you with me? It might be a fan of Cristiano Ronaldo or a fan of Lionel Messi. It might allow Cristiano Ronaldo to pass and not allow Lionel Messi to pass. Basically, it might allow Manchester United fan to pass and not allow Chelsea fan to pass. That is what is meant by what selective. It selectively allow. It doesn't allow all material to pass. There are some basic material that can actually pass. Again, osmosis is the movement of water molecules from a region of lower concentration to a region of higher concentration through a semi-permeable membrane. Are you with me? So it is sufficient to know that osmosis is a special form of diffusion. Note, osmosis is a special form of what? Diffusion. Osmosis is a special form of diffusion. Now, what are the factors that are affecting osmosis? I'm going to look at that. The factors that are affecting osmosis. Factors that are affecting osmosis. Factors that are affecting osmosis. The factors that are affecting osmosis. Factors that are affecting osmosis are there needs to be the presence of a solution with higher concentration. It needs a solution of higher concentration. Are you with me? As well, it needs a solution of lower concentration. This is a solution of higher concentration. This is a solution of lower concentration. It needs a semi-permeable membrane as well. Are you with me? It is a solution of higher concentration, a solution of lower concentration, and a semi-permeable membrane. So let's take a look at this. A cell as an osmometer. A cell as an osmometer. Now let's look at cell as undergoing osmosis. Now there's no way you talk about a cell as an osmometer without looking at this basic fact: hypertonic, hypotonic, and isotonic. Hypertonic, hypotonic, and isotonic. Now we're looking at these key root words: hypo and iso. Are you with me? Hypertonic. Now you might be asked a question. Can you please explain to me the phenomenon of a cell being placed in an hypertonic solution? Or what will happen when a cell is placed in an hypertonic solution? Now there's no way you would get to define that or get to explain that without understanding the concept of what? Hypertonic. Now hypertonic deals with what? Solution. Hypotonic deals with solution. Isotonic deals with what? Solution. Now what is an hypertonic solution? An hypertonic solution is a solution of higher concentration. An hypertonic solution is a solution of higher concentration. It is a solution of what? Higher concentration. Now, if you have a cell such as this, and that cell is placed in a surrounding, and the surrounding has a solution that is hypertonic. The surrounding has a solution that is hypertonic, and it happens that the cell inside the hypertonic solution is hypotonic. What will happen? Now we'll tell you what will happen in a jiffy. Now what is an hypotonic solution? If hypertonic solution is a solution of higher concentration, hypotonic solution is a solution of lower concentration. Hypo talks about low. Hyper is high. Hypertonic solution is a solution of higher concentration. Hypotonic solution is a solution of lower concentration. So now if a cell is hypotonic and it is placed in an hypertonic solution, what will happen? This is what will happen. Basically, if an hypotonic cell is placed in an hypertonic solution, the constituents of the hypotonic solution will move out into the hypertonic solution. The hypertonic solution will draw it out. It will draw everything inside of it. So what will happen to the cell at the end of the day? The cell will shrink. It's just like, for example, okay, imagine me having a pot belly. I am now placed inside a well. Now that well, the solution of the well is hypertonic, while the solution in my inside is hypotonic. So once I get into that solution, hypertonic solution is higher than my own solution. It will draw out everything in me. Now instead of things to be coming into me, it will be going out. It will be going out until I shrink and probably die. So that is the effect of an hypotonic cell being placed in an hypertonic solution. Now, however, how about when we have an hypertonic cell? The cell is hypertonic and the solution of the surrounding is hypotonic. What will happen? And now it happens that I'm placed inside a well. The solution inside of me is hypertonic, while the well is what? Hypotonic. What will happen? I will draw in water from the well. Water will come into me instead of what I have inside me going into the well. So now, looking at it that way, I might get to a point I will now have pot belly. Are you with me? So basically, a cell being placed in an hypertonic solution or an hypotonic solution needs to be clearly understood. When a cell is placed in an hypotonic solution, it will draw in water. When a cell is placed in an hypertonic solution, it will lose water. So basically, hypertonic solution is higher concentration, hypotonic solution is lower concentration. Isotonic solution means the solution of the cell and its surrounding are the same. The solution in me and the solution of the well, they are equal, they are the same. So nobody is drawing in water, nobody is losing water. So can I just take it again? Hypertonic solution is a solution of higher concentration. Hypotonic solution is a solution of lower concentration. Isotonic solution talks about the solution of the cell and the surrounding being the same. So can we just move on now? We'll be looking at the differences between diffusion and osmosis. But before we get there, so a few questions will pop up on your screen. Try as much as possible to attend them. Okay, so now the question is this. What are the basic differences between diffusion and osmosis? We've established the fact that diffusion is the movement of ions or molecules from a region of higher concentration to a region of lower concentration until they are evenly distributed. We've established the fact that diffusion has to do with liquid and gases. We've also established the fact that osmosis is the movement of water molecules specifically from a region of lower concentration to a region of higher concentration through a selectively or differentially permeable membrane. I said whatever allows certain substances to pass only is said to be selective. So now what are the basic differences between diffusion and osmosis? Okay, basically, so one of the major differences between diffusion and osmosis is that diffusion occurs basically in liquid and gases. Diffusion occurs basically in liquid and gases, while osmosis occurs in liquid only. Diffusion occurs in liquid and gases, while osmosis occurs in liquid only. Another point is this: for diffusion, there is no need for a selective permeable membrane. There is no need for a semi-permeable membrane. For osmosis, there is a need. Are you with me? I said they occur in liquid and gases. In osmosis, it occurs in liquid only. Yet there is no need for a selective permeable membrane. And for osmosis, there is a need. And lastly, diffusion occurs in living and non-living organism. Diffusion occurs in living and non-living organism. Osmosis occurs only in living organism. Now can we just take it again? Diffusion occurs in liquid and gases. Osmosis occurs in liquid only. Osmosis needs a semi-permeable membrane, a membrane that is selective or differential. Diffusion has no need for that. And lastly, diffusion occurs in living and non-living organism, while osmosis occurs only in living organism. And so now we're going to be looking at plasmolysis, by extension hemolysis, a difference between plasmolysis and hemolysis, turgidity, and flaccidity. Can we just go there now? Now class, what is plasmolysis? What is plasmolysis? Now whenever you hear the word plasmolysis, what should readily come to your mind is to plasmolyze. Plasmolysis simply means to plasmolyze. Now what does it mean to plasmolyze? To plasmolyze means to shrink. An action leading to shrinkage. That is plasmolysis. An action leading to shrinkage. An action leading to shrinkage. Shrinkage simply means the cell losing its attachment to the cell wall. Now, this is a plant. This is the cell in the plant. This is the cell in the plant that is attached to the cell wall. Now, if the cell is placed in a solution, now it happens that the concentration of the solution is hypertonic compared to the concentration of the cell. Are you with me? The concentration of the cell itself is hypotonic. It is low in concentration, while that of the surrounding is what? High in concentration. Now what will happen? This is what is going to happen. Whatever is present inside the cell is going to be drawn by the surrounding. Whatever is present inside the cell is going to be drawn by the surrounding. So this will happen. Whatever is present inside the cell will now be going out gradually. It will be going out gradually. A time will now come. This particular cell will now leave this place. It will be like this. It's shrinking. It's shrinking. A time will come again. It will go very low, shrinking like that until it will die. Are you with me? It will shrink totally. So plasmolysis can basically be defined as an excessive loss of water from the plant due to the fact that it is being placed in an hypertonic solution. This then leads to the shrinkage of the cell from the cell wall. Are you with me? Ultimately, it ends in death. The process by which a cell loses water when placed in an hypertonic solution, leading therefore to the shrinkage of the cell, is known as plasmolysis. Again, when a particular cell is placed in a solution that is hypertonic to it, it loses every of its content to the surrounding. By implication, the cell membrane will shrink away from the cell wall. By implication, the cell itself will shrink away from the cell wall, and ultimately the cell will die. Ultimately, the cell will die. So that's what we call plasmolysis. Plasmolysis will only occur when a cell is placed in an hypertonic solution. Plasmolysis will only occur when a cell is placed in what? An hypertonic solution. A solution in which its concentration is greater than that of the cell content. So now we're going to be looking at hemolysis. Hemolysis. The word hemolysis, what does it mean? That's what we're going to be looking at now. Now it's sufficient to know that plasmolysis is the opposite of osmosis. Plasmolysis is the opposite of what? Osmosis. Now basically, hemolysis, what is hemolysis talking about? Now look at this word, H-A-E-M-O, hemo. Now when you hear the word hemo, what comes to your mind? Now if you've been a studious student, as someone who takes time to study, you would have known that hemo is talking about something that is related to hemoglobin. Hemoglobin. Hemo is talking about something that is related to what? Hemoglobin. Are you understanding that whenever the word hemoglobin is mentioned, it's talking about what? Blood. And whenever the word blood is mentioned, it's talking about red blood cell. Are you with me? So hemolysis specifically affects red blood cell. Hemolysis specifically affects red blood cell. Now red blood cells are also called red blood corpuscles. Corpuscles. Red blood cells are also known as red blood corpuscles. Now what is hemolysis therefore? Hemolysis is the process about which a red blood cell is placed in a solution that is hypotonic to it. Take note, for plasmolysis, the cell is placed in an hypertonic solution. It shrinks as a result of that fact. Are you with me? Because the surrounding is drawing everything inside of it. So it's just shrinking, it's just shrinking, it's just shrinking. But for hemolysis, the reverse is the case. Hemolysis deals with what? Red blood cells specifically. When a red blood cell is placed in a solution that is hypotonic to it, it means the solution of the surrounding is low in concentration, while that of the cell is what? High in concentration. When a cell is placed in that kind of solution that is low in concentration compared to its own concentration, do you know what happens? It will draw in water from its surrounding. A time will come where the drawing in of water will now stretch the cell itself. The cell will now become turgid. The cell will now become turgid due to the what? Inflow of water. The cell is taking in water, is enlarging, is enlarging, is enlarging, is enlarging, is enlarging, is enlarging. It will get to a time the cell will burst. Pow! When the cell bursts, the cell dies. Are you with me? The cell will burst, the cell will die. Are you with me? The process by which the cell bursts due to the excessive intake of water from the surrounding as a result of the fact that the cell is placed in an hypotonic solution, so to speak, is what we call hemolysis. Take note, hemolysis affects red blood cell. It occurs when the cell is placed in an hypotonic solution. What happens? Excessive water flow into the cell. The cell gets what? Stretched. The cell develops pot belly, as the case may be, and the cell eventually bursts. So that is what we call hemolysis. So now we're going to be looking at differences between plasmolysis and hemolysis. Now follow me closely. Let's quickly look at that. Plasmolysis and hemolysis. Okay, so plasmolysis basically occur in plant cells. Plasmolysis occur basically in plant cells. Hemolysis, as I've said earlier, occur in red blood cell. Basically, it occur in red blood cells or red blood corpuscles. Plasmolysis occur in plant cells. Hemolysis occur in red blood cells, basically. Now let's look at another point. Now plasmolysis occur because the cell is being placed in what? In a hypertonic solution. In a hypertonic solution. Now what happens to hemolysis? Hemolysis will occur when the red blood cell is being placed in a hypotonic solution. In a hypertonic solution for plasmolysis, in a hypotonic solution, in a hypotonic solution for what? Hemolysis. Don't forget, in an hypertonic, here the cell shrinks, here the cell bursts. Why does the cell burst? Because the cell has taken excessive water, so it bursts. Boom! Here the cell shrinks because the outside is calling, give it to me, give it to me, give it to me, so to speak. So that is what we need to know about it. So now basically quickly we'll be looking at turgidity and flaccidity, but before then I still need to establish a fact. Plasmolysis deals with plant cell, hemolysis deals with red blood cell. In plasmolysis, the cell is placed in an hypertonic solution. In hemolysis, the cell is placed in an hypotonic solution. Don't forget. Lastly, for plasmolysis, the cell shrink. For hemolysis, the cell bursts. Thank you very much. Now we're going to be looking at turgidity. Quickly, let's go. Now, what is turgidity? What is turgidity? Whenever you hear turgidity, what comes to your mind should be turgid. 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 Now, turgidity is a process or it's a phenomenon characterized by the inflow of water from the surrounding. When the cell is placed in an hypotonic solution, by now you should be able to establish the fact that whenever a cell is placed in an hypotonic solution, it draws in water. Whenever a cell is placed in an hypertonic solution, it loses water. So turgidity can only be seen when a cell is placed in an hypotonic solution. The cell draws in water. It gets to a point the cell itself gets enlarged, gets stretched, it gets filled up with water. At that point, you say the cell is turgid. When the cell, which is initially like this, takes in water, it gets stretched. So we say here the cell is what? Turgid. So turgidity is a process by which a cell becomes turgid. A cell becomes turgid when it is placed in an hypotonic solution. It draws in water onto itself. It gets enlarged, stretched. It gets bulky. So in that part, at that particular point, we say the cell is what? Turgid. We say the cell is what? Turgid. So now quickly let us look at flaccidity. What is flaccidity therefore? What is flaccidity? Or what is flaccidity? So flaccidity is a process by which a cell becomes flaccid. To become flaccid simply means you're saying the cell is becoming flat, so to speak. The cell is becoming flat. How is this possible? It is however possible when a cell is being placed in an hypertonic solution. As I said earlier, when a cell is being placed in an hypertonic solution, it loses water. So imagine me now being filled with water. I lose all my water to the surrounding. Initially, I was like this. I now lose all my water to the surrounding. What happens? I will become like a flap. I will become flaccid. I become like a flap, flaccidity. Are you with me? So can we just take it again before we call it a day for this particular class? So I said diffusion is the movement of ions or molecules from a region of higher concentration to a region of lower concentration. Just like when you are spraying insecticide or your perfume, the point of spraying is very, very high in the smell. You're like, wow, this is awesome. Especially those perfumes for northerners, Malan perfume, so to speak. They are very, very conk. You say, wow, this is awesome. But when it gets to some particular part of the room, you notice that the smell is not so strongly perceived like where you actually sprayed it. But a time will come, it will not be evenly distributed, or just like even when your body undergoes fatting, basically. But I hope you know that fatting occurs when protein substances are broken down. So don't be ashamed of your part. Embrace it. And so basically, we look at the factors that are affecting diffusion. I said the nature of materials, whether solid, liquid, or gases, the molecular size. I said diffusion affects greatly gases and liquid. I said molecular size, the smaller the size, the greater the rate of diffusion, the bigger the size, the lesser the rate of diffusion. I said concentration and temperature affect diffusion. As both are increasing, diffusion is increasing. As both are decreasing, diffusion is decreasing. Then from there we look at what osmosis. Osmosis is basically the movement of water molecules from a region of lower concentration to a region of higher concentration through a semi-permeable membrane or a selective or differential permeable membrane. Then we look at the difference between diffusion and what? Osmosis. Remember I said osmosis is a special form of diffusion. Osmosis deals with water molecules. Diffusion deals with liquid and gases. Are you with me? Are you with me? In diffusion, there is no need for a semi-permeable membrane. In osmosis, there is a need. Diffusion occurs both in living and non-living organisms. Osmosis occurs only in living organisms specifically. Plasmolysis, I said, will occur when the cell is placed in an hypertonic solution. The cell loses its content, therefore the cell shrinks and the cell dies. Hemolysis here, I said earlier, hemolysis affects red blood cells specifically. When the red blood cell is placed in an hypotonic solution, it draws in water, it gets to a particular point, it develops fat belly, and it bursts. Whoa! That is it. Thank you very much. Now we have difference between plasmolysis and hemolysis. As I said earlier, hemolysis for red blood cell, plasmolysis for plant cell. Plasmolysis occur in an hypertonic solution. Hemolysis occur in an hypotonic solution. The cell shrinks in plasmolysis, the cell bursts in hemolysis. I said turgidity simply means a process or a phenomenon characterized by the placing of a cell into a solution that is hypotonic to it. What happens? It draws in water and the cell enlarges. The cell becomes turgid at that stage. Flaccidity is the process by which when a cell is placed in a solution that is hypertonic to it, the cell loses its content, its water. It becomes like it therefore becomes like a flap. It becomes flaccid, flat, so to speak. So with this, we have come to the end of this wonderful class. Remember, a question will pop up on your screen. Try as much as possible to make attempts. It's been a wonderful time out with you. Peace out.